Hello, everyone, and welcome to another educational session from New Brain Nutrition and Eat to Be Nice. Today, we're talking about how stress in early life may influence development in healthy infants and how the microbiota may play a role in this. We're going to be speaking today with Dr. Carolina de Weerth, and we'll show you our photos here. On the left, Dr. Carolina de Weerth. She's the chair of the Developmental Psychobiology and also professor at the Faculty of Medical Sciences at Donders Center of Medical Neurosciences and the Donders Institute for Brain Cognition and Behavior. And my name is Jonathan Marks. I'm president of Inkwell Medical Communications and also the dissemination director and education director for New Brain Nutrition and Eat to Be Nice. So with that introduction, I'll let Carolina take it away. Here you go.
So hello, everybody. Today, I'd like to do this presentation revolving around two topics, dividing my title in two. So how may stress in early life influence development in healthy infants? And how may the gut microbiota play a role in the link between stress in early life and child development? So first, how may stress in early life influence development in healthy infants? To answer this, we first want to know what type of stress are we talking about. And I think it's easy to look at some photos that I put together to think whether we consider this early life stress. For example, this is a child living in poverty. Would we consider this to be early life stress? Or a child that's living on the streets, is this early life stress? Or children that are psychologically abused by their parents, would this be early life stress? And finally, if a child goes through situations or environments that are very insecure and very frightening, would we consider this to be early life stress? I think we all agree, and the literature has shown over these last several decades, that extreme poverty, fear, abuse, and neglect constitute forms of early life stress. And when we're talking about these types of early life stress, we're talking about chronic stress, so stress that goes on for a longer period of time, from which the child can recover insufficiently. So he doesn't have the coping capacities to recover from this stress that is very continuous and very long. And usually early life stress is known to have a large impact on later life, psychological or physical health. This is related to the phenomenon that is known as developmental programming. And by developmental programming, we mean long-lasting or even permanent effects on development resulting from interactions of an organism with the environment early in life. So early life is a sensitive period, a period of great plasticity in which the environment has the opportunity to put a mark on development. And that's why we call it developmental programming. Its influences are long-term and important, and they may range from influences on metabolism, on cell functioning, to the functioning of the brain and behavior. But these were very severe examples of early life stress. What about milder forms of stress in early life? The types of stress that maybe community healthy infants come across in their normal daily lives. For example, if your mother during pregnancy works and has a busy job and some worries during her job, could you consider this early life stress? Or if your mother does pursue some more extreme physical activities during pregnancy, would this be early life stress for the fetus?
That's quite a bit of weight that woman is lifting.
Yes, that is true. That is true. Or maybe if your parents are not that talented in caregiving for a young infant that tends to cry a lot, is this also early life stress for the baby? Can we consider this maybe a less ideal environment with some type of mild stress for the baby and the parents, of course? Or if you go to childcare, center-based childcare very early in your life and have to compete for the attention of caregivers that have to take care of several children at the same time, Is this also early life stress?
These are very good questions.
Yes, and finally, this very actual or very question from nowadays, which is if children have to compete for the attention of their caregiver with this phone in the hands of their caregiver, is this a form of early life stress? We could, of course, answer all these questions saying, well, stress is normal. It's even adaptive. We need the stress system to survive. It's necessary to be able to adapt to the challenges of everyday life. Children need to be confronted, even the fetus, of course, needs to be confronted with differences in stress levels in life. And it's only maybe a positive adaptive experience that prepares the individual to better adapt to his environment later on. Or in other words, how bad can a little bit of stress be in your life? If we look at the fetus, we could say, well, isn't he safe in the womb? Isn't the fetus completely protected in the uterine environment? And of course, if we think of things such as alcohol or smoking, that would be negative for the fetus. But if the mother is just, exercising a little bit more fanatically or a little bit worried because of her busy job, is this really so bad for a fetus?
Or emotional distress if there's tension in the couple?
Yes. If you have, for example, relational problems or psychological stress or maybe mood changes, we know that many pregnant women have mood changes, but you could reason this is happening in the mother's head. Why would it affect the fetus? And if we think of the early environment postnatally, we could say, okay, a little bit of challenges in a little bit of a chaotic caregiving environment. If there's enough food, if there's enough love around, well, what doesn't kill you makes you stronger. Kids, they're pretty resilient and they can grow even though there's a little bit of adversity, as long as it's not, of course, very harsh and very serious. We could reason that way. And this leads us to two important questions. Are these variations in early stress in community low-risk samples actually related to development in young children? It could be that these are just normal variations that actually have no relation at all to later development. And if they do have a link with later development, are these variations in early stress in community samples actually related to worse development in these children? And these questions are very relevant for us as a society to know whether we should invest our scarce or limited, you could say, resources in general prevention, or whether we should save the money for better interventions for more extreme cases in which there is real risk of developing physical and psychological problems later on.
Carolina, you really have my interest here. You've asked a lot of questions and I don't know the answers, so I'm waiting for them.
I hope I can help you. Let's see where we go with this. We'll probably end with some questions actually, but so just a note that I want to say here on the side, these examples that I've given of milder stressors in early day life do not mean that I'm implying that mothers shouldn't work, exercise, use childcare, or have a phone. It's not that I'm against these things, but I'm just using them as illustrations of how they could produce variations in stress in the environment for young children and the fetus, of course. So I'm going to start with showing some examples from our own studies that may help us come to an answer for these questions. First, we looked at prenatal stress and anxiety. And by prenatal stress and anxiety, I mean just what you were talking about, Jonathan, the worries that a woman may have because of relationship problems, because of mood swings. So all the anxiety, depression, and maybe daily hassles, life events that happen to her during the pregnancy. I'm going to call that prenatal stress just to make it easier to talk about this, but it implies all these different types of symptoms. And what we did in our own studies is related to the child's health and the child's stress system. Mm-hmm. And I want to put the accent on relationships or associations that we studied because these were not, of course, randomized control studies. We did not stress women during pregnancy. So the results I'll be showing are just associations and that they are not causal relationships. The first study is the relation between the maternal prenatal stress and health in the infant's first year. We here had monthly health interviews with the mothers, and we noted all the symptoms in these children. These children that participated in our studies, or the mothers rather, were quite healthy mothers that were relatively highly educated. So we did not expect their children to have many illnesses in the first year. So we asked for all kinds of symptoms, from coughing and fevers and runny nose to more serious health complaints, of course. And we found that if the mother was more stressed during pregnancy, the child actually was reported to have more general complaints and illnesses, skin illnesses, and respiratory illnesses. And he also had more antibiotics prescribed during the first year of life. So this is a very interesting association. We also found that the associations persisted for general and respiratory complaints, and we studied it until the age of 6 years. We're still following these children, so we can look in the future to see whether these associations still are there.
Now, why can't I say, Carolina, just hearing this little bit of data, why can't I say that prenatal stress causes poorer health? You're saying it's just association, but doesn't it seem to make sense that prenatal stress, maternal stress would cause poorer health in an infant?
Well, there are all types of mechanisms that could be in place that could be what you're saying. So, for example, if a mother is stressed, her immune system may be affected and working less well. She may be transmitting less immunity to her child, and this makes the child more often sick during the first year. That could be a causal explanation for these findings. But we did not study this mechanism in its whole, and it could be explanation that there are third factors that are actually causing both. For example, a mother that is stressed prenatally may be more stressed postnatally, and she may be caring for her baby in a less adequate manner, so less high quality manner, and this may produce stress in the baby, and this may depress the child's immune system, and that's why he's getting sick. So as you see in this example, it's not the prenatal stress that is causing the illnesses. No, the prenatal stress is related to the mother's behavior postnatally, which in turn is related to the poor health in the baby. So they can be other mechanisms and not direct causal mechanisms that we're talking about here.
Thank you.
So another example of a study that we did was looking at how maternal stress during pregnancy was related to how a baby habituates to a repeated stressor at 9 months of age. This is a very ecologically valid example, I think. I really love this study because I think it imitates a very natural situation. What we did is have the mother in their home leave for an hour, and we had the baby taken care of by a babysitter. And this babysitter was, by the way, very boring. She just read a magazine, except if the baby cried, then she, of course, comforted the baby. But if not, she was just boring, because this is what produces a little bit of stress in the infant, and that was what we were looking to do. And we were interested in seeing whether the infant adapted to this situation over the weeks, whether he or she habituated. We saw that over the weeks there was less crying over the separations, and that there was lower levels of the stress hormone cortisol. The stress hormone cortisol can be measured very easily in saliva of the baby. So it's non-invasive, and you can do that very easily. But in the infants where the mothers were prenatally stressed, we did not observe this reduction in the stress hormone cortisol over the weeks. This could, of course, have different meanings. It could be that if we did a fourth separation a week later, these separations were separated by 1 week. If we did a fourth separation, then maybe the babies would have adapted, even the babies of prenatally stressed mothers. But it could be also, and that's an alternative explanation, that these children are more sensitive to stress and are less able to regulate their stress system and get used to or habituate to repeated stressors. In turn, this could be indicating that these babies are more prone to chronically elevated levels of cortisol, which is a form of chronic stress that could be affecting their bodies negatively. These are just speculations. These are interpretations of the results. And then some examples of our postnatal studies. So what about the postnatal environment? Stress in the postnatal environment in these healthy babies. We related them to sleeping arrangements and center-based childcare to child outcomes. The first thing we looked at is co-sleeping, or in other words, room or bed sharing versus solitary sleeping. In a longitudinal study, our BIBO study, we had 30% that slept solitarily in their own room from day one during the night, and we had the rest of the children that slept either in the parents' room or in the parents' bed. We actually only had 5% of children that slept in the bed of the parents, and taking these out of the analyses did not affect the results. So what I'm going to be talking about mostly is room sharing. And by room sharing, I mean sleeping in the same room as the parents on a different surface, just like in the photo. We looked at these sleeping arrangements during the first 6 months of age, and we related them to cortisol reactivity. Cortisol reactivity is the heightening of the hormone cortisol in reaction to a stressor. It's a very normal physiological reaction that we all have when we're stressed, and it usually consists of a temporary increase in the hormone cortisol, followed by recovery, a decrease in the hormone again. And these three situations that are in the slide are known to produce a cortisol reaction in infants. The first one is a bathing situation by the mother, just putting them in bath, taking them out of bath, produces an uncomfortable sensation, cold, and getting dressed again. And usually infants show a reaction. A vaccination is a pain stressor. And at 12 months of age, you have a maternal separation, which is the Ainsworth strange situation, which is a lab protocol that is done to measure attachment or to assess attachment. This also produces an increase in the hormone cortisol. So what we found is that if the babies had had more room sharing in the previous months, their reaction to the bathing situation showed lower increase in cortisol. There was no difference between the groups of solitary sleeping and room sharing infants for the vaccination, and there was a lower cortisol reaction to the separation from the mother at 12 months. These findings could be indicating that sharing the room with the parents permits the infants to develop maybe a more flexible or more regulated cortisol system, in that they do not react with exaggerately high cortisol levels to what you could call daily stressors, such as a short maternal separation or, a daily bath, which they are used to having every day. And it could mean that babies that sleep solitarily are perhaps less well regulated because it takes them a little bit more effort to get the parents to come and calm them down and settle them down again at night than the babies that are room sharing. And this produces changes in their cortisol physiology that leads to higher reactions to stressors in their lives. But we do not know. These are the mechanisms that need to be studied in further research. And finally, we looked at childcare, and children in the Netherlands start childcare very early on at 3 months of age. And we found that if you look at their cortisol levels in childcare compared to their home levels, that they have higher cortisol in childcare in the mornings than at home. And this continues for many months after starting. Also, if they spend more hours in childcare, they have more respiratory and general illnesses. This could be possibly explained by contagion. These illnesses, respiratory and general, are often illnesses that are obtained through contagion from other children. And we finally found that if they had more concurrent arrangements, they had more skin illnesses. Concurrent arrangements, by concurrent arrangements, I mean that children, next to going to center-based childcare, also had other childcare arrangements. In the Netherlands, it's very common to only send your child for 1 day a week or 2 days a week to center-based childcare, and to have grandparents from your husband's side and your own parents to take care of the baby, for example, for 2 other days. And this means that there are more concurrent arrangements. These children may be having a little bit more stress, at least that's our theoretical explanation or possible explanation for these results, because childcare is a very busy environment. There are a lot of changes in childcare teachers in the Netherlands because many of them also work part-time, so they do not always get the same childcare teacher. And this could be producing more adaptation or challenges for the baby, and that's why maybe they have more skin illnesses, which are known to also be triggered by stress.
I was going to ask about skin illnesses. It's the second time you've mentioned it in your slides. Could you explain a little bit more about the connection between stress and skin illness?
So people that suffer from atopic dermatitis or eczema are known to have worsening of symptoms by stress. And this is the idea that we have, that maybe there's more being asked of children that have more concurrent arrangements, as they have different challenges to adapt to different caregivers through the week. And this may produce stress, and this may make them more vulnerable for skin complaints such as dermatitis or rashes.
Mm-hmm. Thank you.
So coming back to our 2 important questions, are these variations in early stress in community low-risk samples actually related to development in young children? I would say the answer is yes. We do find relations between these factors in community low-risk samples. And the second question is whether these variations in stress in community samples are linked to worse development in these children. And that is more difficult to answer. The problem is that we do not have enough longitudinal studies, or long enough longitudinal studies, on normal population variations. So there have been studies on orphanage children or children from foster care, children that have had abuse or neglect, so severe forms of harshness in early life. But we do not know enough whether it leads to more physical and psychological complaints later on in adult life, for example. But from animal studies, we do know that these relations are causal, that even more subtle forms of stress can be related to worse development. Again, it's difficult to answer this because what is worse development? Sometimes effects can be negative in the long run, but they can be adaptive in the short run. So that's another problem. But we also know from natural disaster studies in humans, such as earthquakes or other disasters, that having stress early in life can lead to, or is related to, things like depression later on in adult life. Also, early life stress is a continuous measure. So we can talk about severe forms of early life stress and mild forms of early life stress, but it is a continuum. And it also very much depends on the individual. Some individuals are more vulnerable for stress or have fewer coping skills for stress. So it also depends on the interaction between the environment and the specific individual, of course.
Carolina, I have 2 questions. The first
Yes?
is Eat to Be Nice conducting more longitudinal studies at this point?
That's a good question. And yes, I know from our work package at least that there are several longitudinal studies going on. There are also very large epidemiologic longitudinal studies, which I think will produce very interesting results. I'm not sure that there are many longitudinal studies in young children or in like our longitudinal studies. We have at least 2 in which we will be collecting data on impulsivity, on aggression, on regulation behavior in toddlers and also in adolescents.
Mm-hmm. So Eat to Be Nice is really contributing to the scientific knowledge here. That's the purpose of the whole project.
Yes. We will for certain contribute with all the research that is being done at the moment to answer these questions, for sure.
Great. And so we will keep you updated on our results as we get them, as they become more conclusive. My second question, Carolina, is I'm sitting here, as I think many parents are at this point in the presentation, saying, oh my God, what have I done to my child? And, how much stress did I put on my child, either knowingly or unknowingly? Are any of these, it seems like stress, as you mentioned before, is kind of a natural part of life. A child can't get everything it needs whenever it needs it. But I'm just wondering, are there answers for this? Are there ways we can ameliorate the stress that will, make the consequences not so bad in later life? Do we know about those yet?
Well, the human life is a very long life compared to, for example, lab rats. And we know that a lot of things happen later on in life that compensate for example, the effects of stress in early life. What we do know is that early life is a very sensitive plastic period. So what happens in early life, you could say, has more weight than what happens, for example, in middle childhood, often, just to put it very black and white. But of course, there are very many ways of compensating for things that maybe did not go as well, or where you maybe had mild stress, for example, during pregnancy. And this can be very well compensated by high quality caregiving later on, for example. That could be an example. I'm not saying that this is the case, but we know from many studies that if you look at different environmental influences in different stages of life, you will find different results. So to put it simply, I don't think parents should worry, because life is very complex. A human being is a very complex being in a very complex environment, and there are many, 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 many factors affecting development. And for example, room sharing is one factor, but there are so many other factors that will be maybe much more important, such as, for example, the type of caregiving that parents give, or breastfeeding, or diet, exercise. There are so many things, social relations with grandparents, with friends. There are many, many factors that will influence life. So I don't think parents should worry, or, I think we all start wondering about what we did with our own kids when we hear these stories, but you have to remember that they are very, very small pieces in a huge puzzle, which is a human life.
Thank you. I'm breathing a little bit more easily right now.
Great. You should. It's nothing to worry about. And by the way, things like co-sleeping, for example, that room sharing, I've had people say, well, I couldn't sleep with my baby in my room. The World Health Organization recommends 6 months of room sharing because of SIDS, of sudden infant death syndrome, to prevent that. But many parents will say that they can absolutely do not sleep with a baby in the room because it makes too many noises and they awake and they have very low sleep quality. And I think that if this is the case, then maybe you will be a much better parent if you're well slept and the baby sleeps in his own room and you have slept well and you're in a very good situation to take care of the baby in a very high quality manner during the day. So not everything is good for everybody. That's what I want to say. Yeah.
Got it. Thank you.
So the second topic of the lecture is how may the gut microbiota play a role in the link between stress in early life and child development? What is this gut microbiota? And before I go into that, I'm not going to talk a lot about the gut microbiota in itself. We know, of course, and we've known for decades that it has a very important role in our health, in digestion of food, of vitamins, or the absorption of vitamins, in our immune system development. But what I'm going to be talking about now is the specific relation that it may have with stress in early life.
And I will add here that we have other educational sessions which go into a lot of detail about gut microbiota, so you can learn about that in those sessions. So go ahead, Carolina.
Yes, exactly. So I won't be telling you the basics about gut microbiota, just in relation to the HP axis development. And the HP axis is a hypothalamus pituitary adrenal axis, which is the main stress system of our body and produces as an end product cortisol, which is the stress hormone that I've been talking about a lot, and which has a very important impact on our body. That's why it's studied so much. What I wanted to say here is that during pregnancy and infancy, the gut microbiota develop, and they have, during the colonization of the infant gut, they play a very important role in the development of the brain and also specifically the HP axis. So in green, you can see that they have an important role in the immune function, in microglia control, in catering for the brain's metabolic demands. So the microbiota are essential for our healthy brain development.
And just for people who don't know the technical terms, what is microglia?
Microglia are immune cells of the brain. So they have an immunity role in the brain itself.
Okay, and HPA again stands for?
Hypothalamic pituitary adrenal axis, and it's the whole stress system. So the stress system starts in the hypothalamus. When you're stressed, that's where you get the first reaction, and a signal goes to your pituitary, which again signals to your adrenal cortex, which will produce cortisol. And then cortisol goes through your whole body and produces different activities. And there's, of course, other systems, stress systems that produce adrenaline, noradrenaline, or epinephrine, norepinephrine that have to do with stress. But just to focus now on the axis and cortisol, we know that when you're stressed, it is activated more than usual. And when you have physiological stress in your body, this produces immune changes, neurochemical changes, and physical changes. For example, your gut motility may be affected when you're stressed. And I think a lot of people will recognize this, that during a period of stress, your gut is not functioning as it should. A lot of gut complaints appear when people are chronically stressed. So that's the red part of the graph. Yes.
I just wanted to comment on the chronic stress. These systems that Dr. de Weerth is talking about are normal defensive systems for your body, so that if you encounter a dangerous situation, you do want to have a stress reaction so you can react quickly and you can, flee if you need to. But I guess if these are chronic, this can become somewhat debilitating.
Yes, because the stress system is such an essential part of our survival, thanks to the fight and flight reaction and to other bodily functions that are kept going thanks to the stress system, that we would not be able to survive well without them. But the problem is if it's activated for too long a period and if we lack the coping mechanisms to recover from these periods of stress, physiological or mental coping mechanisms. So in this case, in the graph, what I'm talking about is that when these stress effects happen in the body and it becomes more negative for the body, it's not just a transient, very quick stress reaction, but kind of a larger or longer stress reaction, it may affect the development of the gut microbiota. It's very important that the gut is colonized early on in a healthy way and that the gut microbiota develops to become a balanced colony of bacteria in our gut. When this does not happen, there are imbalances in the gut microbiota, and the gut microbiota itself may produce metabolites that are not very good for our body, but also the normal functions that are maintaining brain development will be disrupted. And that's why the red line is crossing the green line. So the normal healthy development of the brain may be affected by imbalances in the gut microbiota. This in turn can lead to the HPA axis being dysregulated. And by dysregulated, I mean reacting too high to stressors or too low. At a certain moment, it can actually be a hyporeactivity to stress. The gut microbiota may become dysbiotic. You can see dysbiosis, and dysbiosis is a fancy word for saying unbalanced. It's an ecosystem. You can see it as a jungle where every item in the jungle has its role. And when there is dysbiosis, this becomes dysregulated, unbalanced. The tight junctions between the cells of the gut become more loose. You get what is called the leaky gut. Bacteria or the products of bacteria can come into the system and produce low-grade inflammation. The modulation of the central nervous system of the brain, for example, can be affected. And in turn, all these effects can lead to alterations in our behavior, health, emotions, and cognition during childhood and adulthood. This is the theory behind how the gut microbiota and the HPA axis in development can affect later outcome in adulthood and also in childhood already.
And we know from the other presentations that you'll learn about with New Brain Nutrition that there is a direct connection between gut microbiota and the brain in the what is called the gut-brain axis. So, I think we used to think of our brains as totally separate from the rest of our systems, but they really are one system and they're very reactionary with each other.
Yes, I think you're making a very good point there. We should see the gut microbiota as a new organ, as if it were a liver or a kidney. It's like a new organ in our body that we've just recently discovered has so many functions that we had not even suspected, and that is in very close communication with the brain. Some examples from our own studies on gut microbiota. We were curious to know whether prenatal stress and anxiety was related to later infant gut microbiota. So we related both of these, both reported maternal stress as well as cortisol in the mother. You would say, why measure cortisol in the mother? If she reports that she's stressed, depressed, anxious, that's enough. But by also measuring cortisol in the mother, we have two different sources of information on the stress levels of the mother. And these sources are not always correlated. This is contrary to our intuition. We would say that when we are feeling very stressed, it's natural for us to have high levels of cortisol, but this is not the case. There are studies also doing observations on people, and they have found that behavior that looks completely stressed is not related to high levels of cortisol always, and the other way around is the case as well. People can look very calm and have very high levels of cortisol. So we did both measures, both sources of stress were measured in the mothers, and they were related to infant intestinal microbiota. And here you can see different relative abundances of the bacterial groups. So this is a kind of a complex slide, and I'll guide you through it. We looked at different clusters of bacteria, you could call them bacilli, proctobacteria, bacteroidetes, clostridia, and actinobacteria, and those are just fancy names for different types of bacteria. And we looked at the relative abundances in the fecal samples of the infants. We took 5 fecal samples in the first 100 days. So these represent a little bit the colonization of the gut of the infant, because the infants are born with almost sterile intestines, and the bacteria colonize the gut very quickly and establish more or less stable colonies, or you could say ecosystem. Well, there is some discussion, but most people say that by age 3, it's more or less stable. So it changes a lot during early development. And what can we see in these graphs between two more or less extreme groups? And I call them extreme groups because we took the children that had mothers with low cortisol levels and low reported stress. So you could say these were all the calm mothers, and the more stressed mothers during pregnancy that had high cortisol as well as high reported stress. So they were, you could say, doubly affected. As you can see in the graphs, the protobacteria are much more abundant in the high stress group. And the protobacteria include species that are gas producing and that are related to pain and to dysbiosis. So it may be not that good to have so many protobacteria. And as you can see in the low stress group, the protobacteria are not that abundant, and they do not change over time either. The actinobacteria, on the other hand, the green surface, you could see that in the low stress group, they increase over time, and these include the genuses of bacteria that are normal and that should be present in the infant's gut. And you can see that in the high stress group, the increase in actinobacteria is much lower than in the low stress group. Because we had also interviewed these mothers on their infants' health, we also exploratorily looked at their health symptoms. And we found that the digestive symptoms, which were higher, so digestive complaints in the infant, were much higher in the high stress group, as well as the allergic symptoms as well. And of course, these infants were only 100 days old, so these are just very, you could say, soft complaints, no diagnosis yet. But it's interesting that it does go in the same line of results as the bacteria.
It's interesting you said that prenatally the infant's gut is sterile. So it's the introduction of mother's milk or food that produces the abundance of bacteria?
In normal infants, there is quite some discussion, but there's pretty good evidence that they actually do already have some bacteria in the gut when they're born. But most of the colonization occurs during vaginal delivery, actually, or most of the bacteria are seeded during vaginal delivery. So children that are born through C-section have a lower diversity of bacteria. It takes them more time to develop a diverse colonies of bacteria or ecosystem in their gut. And they also often have bacteria that are representative of the mother's skin and of nurses in the hospital even. That's why there's actually many researchers that are trying to find ways of inoculating newborns that are born through C-section with the gut bacteria of their mother, because babies are exposed to gut and vaginal bacteria of their mothers during vaginal deliveries. That's how they start their colonization.
Fascinating.
Yes. Yeah, very interesting. It's a natural system, you could say. And other examples that we did was look at bacteria in relation to infant colic. And by colic, I mean babies that cry excessively. And the normal rule of thumb is to say that they cry for 3 hours a day, at least 3 days a week for at least 3 weeks. So that's a lot of crying. And we also looked at the response to the rotavirus vaccine. And the rotavirus is a virus that produces diarrhea and also is linked to infant mortality in many countries in the world. The rotavirus vaccine is given to children, especially in developing countries, but many children are non-responders to this vaccine. So they do not develop the necessary immunity. And I'll show you how that's related to gut microbiota. So first of all, the colic, we had a large group of children, and what we did was compare the small group of children that were very highly colicky to a group of children with very low levels of crying, so very placid babies, you could say. And we looked at 9 samples of gut microbiota in the first 100 days in this study. We found that the babies that had colic, even though we measured the colic at 6 weeks, which is the universal peak in crying, already in the first weeks of life, we found differences in the bacteria between this group of colicky infants with the control group. We found slower colonization and a reduced diversity and stability in the gut bacteria. We found more relative abundance of proteobacteria, and these, as I said before, also include species that produce gas and inflammation. We also found fewer butyrate-producing species, and butyrate is something that is related to less pain in centenarians, it has been related to. So there are some studies that have studied butyrate-producing bacteria in centenarians, so people that are more than 100 years of age, and have found that if they have more butyrate-producing bacteria in their gut, they have fewer complaints of gut pain. Also, butyrate is supposed to be related to brain development in infants and have a positive effect. Another thing that we found is that these infants that were colicky had fewer lactobacilli and bifidobacteria in their gut. And these are the normal bacteria that should be present in large quantities in the infant gut, especially bifidobacteria in the first months of life. And they include species that have anti-inflammatory effects. So these are the good bacteria, you could say. So these different profiles that we found already in the first weeks of life may be indicating that bacteria are actually related to the development or to the etiology of colic in babies. And that is an interesting finding because anecdotally, people have always talked about colic as if it were gut problems, whereas it hasn't been possible over, these last centuries often to find a cause of colic. Some infants are colicky, and there have been many studies trying to find the reason why they cry so much, and they've looked at parenting, and they've looked at allergies, but allergies are apparently only 12% of the colicky cases. And there are many, many studies on colic, but now we're going back to actually the word colic, which means intestine, and apparently maybe at least a part of the cause of colic may be the gut or the gut bacteria.
Are there studies that introduce the right kind of bacteria to babies that can reduce colic?
Yeah, that's a very good question. And there have been large studies, there have been meta-analyses, and it's kind of a baffling answer because actually introducing good bacteria in infants with colic, and these are called probiotics, the good bacteria that have positive effects on health, they have been found to help in breastfed infants, not in formula-fed infants. So that is quite interesting, and the reason why this is the case is not known.
Yeah, that's a puzzle, isn't it?
Yes, it's a puzzle. So that needs to be studied more in the future. There aren't that many trials, of course. But
so one of the things that I love about this Eat to Be Nice project is that we're really seeing a window into the scientific process. Here's what we know, here's what we're studying, here's what we don't know, here's what needs to be studied. And what's wonderful about this project is you're really being invited into this process with our scientists and experts. So I'm so glad you're presenting this information, Carolina, and I'm very thankful to the rest of our presenters for all the information they know that they're sharing with the public.
Yes, it's very interesting. Science in the making. Okay, and the rotavirus vaccine that I had told you about, I'm not going to show you all the data, but there are two different independent studies done in Ghana and in Pakistan, which are countries in which children indeed do not respond very well to the rotavirus vaccine. And they found that the infants that did respond had microbiota that was much more similar to that of the Dutch infants, which are all theoretically responders. So in Western countries, infants all respond to this vaccine, and they found that the microbiota of the children in Ghana and Pakistan that did respond was very similar to that, or much more similar to that of the Dutch infants. So it does seem to be showing this link between early life microbiota and development of the immune system. So then we come back to the more ethical or moral question: do we need to invest in prevention of stress in the community low-risk population? And in my opinion, the answer is yes, because by investing in general universal prevention programs, we can reach many more children than if we only invest in the high-risk population. And you can actually avoid the development of children with high risk, because prevention leads to a healthier start in life, and then in this way you will be developing children that are healthier and that need less societal resources later because they just do not have the physical or psychological problems that others have. So it's, I think, a very good idea to invest. And how can you do this? One thing is that we could do more screening, especially during pregnancy. By screening, I mean to set up routine pregnancy and also postnatal screening. We know that there is a high incidence of depression, anxiety, and stress during the prenatal phase and also postnatally. For example, around 15% of mothers will have postnatal depression, but also 15% of fathers, which is something that is less known. I think that one way of doing this is, of course, you can do a pencil and paper screening test, a depression questionnaire at the midwives or at the obstetricians practice. But I think that it would be much more efficient if we had psychologists that are specialized in pregnancy and early parenting that have one routine talk or visit of all parents that are or pregnant couples or young parents to this psychologist that could be part of the practice of a midwife, for example, and that this psychologist talks about the changes in life that occur when you're pregnant, changes in the relationship, how having a baby may change your relationship, how having a baby can be stressful or not, or just give general information about delivery and parenting. And in this way also screen for parents that may have excessive forms of anxiety or that may be suffering from depression. I think this would be a very efficient way, and I'm sure that a lot of psychologists are very interested in specializing in this fascinating period of transition to parenthood. Another way of doing this is, of course, developing more and better interventions, because once you screen and you discover that a certain part of the population has heightened symptoms, how are you going to intervene to decrease the negative symptoms? The problem with this is that there have been many studies on very different types of therapies for women that have anxiety or stress during pregnancy. For example, that these photographs are only of pregnancy, but we could do the same for young parenthood. The problem is that they do not all work. And that means that a lot of people think that there are no good preventive strategies to prevent anxiety and depression during pregnancy or stress, whereas I think that it's actually the case that one size does not fit all, and that we should be looking at developing more personalized interventions in which each woman, according to her personality, to her situation, and woman, and of course her partner as well, receive the care or the intervention that they need specifically. So the take-home messages are that less severe early life stress appears to be related to health and the stress system development, that while the effects may be smaller, they can still affect health and well-being, and we still don't know enough of how they may be affecting health and well-being in adulthood and even in elderly period of life. And also the intestinal microbiota may be mediating these associations. And this is promising because, as we just talked about, the intestinal microbiota may be modified through the use of probiotics, for example, or through diet. And prevention and interventions may help decrease maternal stress and promote healthy child development. And it's worth investing in these types of prevention and intervention programs because in the end, they have a larger effect, a more positive effect for society than only intervening when the problems have arisen.
So Carolina, the research that you're doing really has been based upon data that's already been collected by public health studies. Is that correct?
No, the research that we're doing is based on data that we ourselves collect. Okay. We do prospective longitudinal studies. We usually start in pregnancy, and our longest study, the children are now 12, 13, but we have some shorter studies.
Great, got it. So in addition to that, we're also doing 4 clinical trials for Eat to Be Nice. And one of them is testing the Mediterranean diet. Another one is testing the elimination diet, eliminating certain foods to see what that effect that has. The third one is probiotics and what effects probiotics have. And the fourth one is vitamin supplementation. These studies are all going to take a couple of years to complete. We're really excited about getting them started, which is happening now, and we'll be able to report to you over time the results. What Carolina has been reporting to us today is what we know from studies that are either finished or are ongoing. This has been really fantastic. It's such valuable work that you're doing, Carolina. What's the continuing work you're going to be doing in Eat to Be Nice?
What we're looking at now is at early life diet, so lactation specifically. We're looking at the composition of breast milk sugars, microbiota in breast milk, cortisol in milk, and also immune factors in milk, and how that is related to behavioral development in children. We're looking into toddlers, and we're also looking at current diet and behavior in healthy adolescents, and at the history of breastfeeding in healthy adolescent behavior.
This is wonderful. And part of the reason the European Union funded the Eat to Be Nice project, when the research is complete, is to end with recommendations that can be implemented in society. And this is what Caroline has been talking about today, is where should we implement, what kind of interventions should we have, so that we can really make a difference in the outcomes of various different health disorders based upon what you eat and your lifestyle. So that's really the purpose of the New Brain Nutrition and Eat to Be Nice project. I want to thank very much Carolina de Weerth. She's the chair of the Developmental Psychobiology and professor of the Faculty of Medical Sciences at the Donders Center of Medical Neurosciences and the Donders Institute for Brain, Cognition, and Behavior. What a great presentation today, Carolina. Thank you so much.
Thank you. Thank you for the opportunity to giving it.
My pleasure. And again, this has been Jonathan Marks with New Brain Nutrition and Eat to Be Nice. And we hope you'll return to learn more from our other educational sessions. Thanks, everybody. Take care.